Hello guys, and welcome back to Future Academy. So today we'll be doing the second part of PCA cycle. So in the previous, we just learned like what is PCA cycle, what are the various steps in PCA cycle. Okay, so let's just summarize this. What we have discussed in the previous lecture. So here you can see this is your PCA cycle, and we have discussed that from pyruvate with the help of pyruvate dehydrogenase you get acetyl-CoA. Now once acetyl-CoA is a 2-carbon. It enters the citric acid cycle by combining with the oxaloacetate, which is 4 carbons. 4 carbon, 2 carbon together will give you citrate, which is 6 carbon. Okay. Okay. Now, from citrate to aconitate to isocitrate. So this step is like which they have shown in the brackets. They are actually very unstable. So immediately they convert into the isocitrate. Okay. So we have got the citrate, aconitate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, and malate. Okay, so all these steps now we will be discussing singly. Okay, so हम लोग यहाँ पे इसके enzymes के बारे में भी discuss करेंगे and how exactly these steps are taking place in the mitochondria. Okay, so this is what we are going to discuss in this. स्मृति को doubt होगा तो please stop me and answer.
Yes, ma'am.
So now citrate synthase. So this is a 2-step reaction and aldol condensation of oxaloacetate and acetyl-CoA. So and 2 carbon, 4 carbon and 2 carbon. It is followed by hydrolysis to each citrate and free coenzyme A. Okay, so the hydrolysis step is not easily reversible. So what do we get? Oxaloacetate along with the acetyl-CoA is going to give you the citric acid. Okay. Now aconitase. Aconitase is the enzyme. Okay. So it is a homodimer, each monomer containing iron sulfur atoms arranged in a cluster called iron sulfur center. So this is important, guys. That's aconitase. So after citrate, aconitase will give you this aconitase. Okay. So this has got your FeS cluster, that is iron sulfur cluster. However, the other hydratase, they lack such iron sulfur center. Okay. Fluoroacetate inhibits the aconitase enzyme. Another point to remember, and thus prevents utilization of citrate by the enzyme. So if you have a fluoroacetate which occurs naturally in the leaves of the certain toxic plant, for example, Dicopetalum simosum, which is a toxic animal that's feed on it. So just एक छोटे से examples है, आपको याद रखना है. So in this slide, what you need to remember is about the citrate synthase is catalyzing the step of oxaloacetate to citric acid. Okay. Then we have got from citric acid to cis-aconitase. Cis-aconitase with the help of the enzyme aconitase. Aconitase क्या है? It's a homodimer and it has got the iron sulfur center. Okay. And fluoroacetate inhibits this enzyme. But आपको इतना ही याद रखना है from this slide. Now, oxidative decarboxylation of isocitrate. तो हमारे पास क्या steps है? Citrate, cis-aconitate, and then you are going to get the isocitrate, right? So isocitrate dehydrogenase will convert isocitrate into a C5 carbon, that is alpha-ketoglutarate. Now, it is an oxidative decarboxylation reaction. इसका मतलब क्या? Decarboxylation. तो यहाँ पे वो जो स्टेप है जहाँ पे आपका CO2 रिलीज हो रहा है, ठीक है ना. डिहाइड्रोजनेज है, as I always said, यहाँ पे आपका NAD plus जो है रिड्यूस होगी in ADH होगा. Always remember. So this reaction takes place in 2 stages. Dehydrogenation of isocitrate to oxalosuccinate, which remains bound to the enzyme. Okay. NAD plus or NADP plus is required as an electron acceptor in the reaction. Now, decarboxylation of oxalosuccinate to alpha-ketoglutarate. So, both these reactions are going to be irreversible and the delta G is negative for this reaction. Okay, clear? Are you able to follow Smriti? If you remember these cycles, then it becomes easy.
Yes, ma'am.
Okay. So now this isocitrate dehydrogenase is 2 steps, one requiring NAD plus as electron acceptor and the other requires NADP plus. Okay, so they are specific. So both the types appear to participate in the citric acid cycle. So आपके पास 2 तरह के isocitrate dehydrogenase है जिसको एक NAD plus लगता है और दूसरे को NADP plus लगता है. Okay. So both the types appear to participate in the citric acid cycle, but the NAD plus specific isocitrate dehydrogenase is predominant. कौन सा वाला आपका NAD plus वाला? So it is mainly present, and it is found only in mitochondria, whereas NADP plus specific enzyme is both found in mitochondria as well as cytosol. And both these enzymes require divalent metal ions like Mg2 plus Mn2 plus for the activity. Okay, so this is about the isocitrate dehydrogenase. So कैसा है, like whenever we talk about TC cycle, nobody is going to ask the citrate के बाद क्या बनता है, right? So they are going to ask about the enzymes, the flow factor, and how that particular reaction is being carried out. Okay. Now, oxidative decarboxylation of alpha ketoglutarate. Okay. So, two successive oxidative decarboxylation steps are the peculiar feature of the citric acid cycle. अब यहाँ पे क्या है? See, if you remember citric acid cycle बोला है ना हमने, तो oxaloacetic plus acetyl CoA will give you what? Citric acid cycle. यानी कि 2 कार्बन हो गए, है ना? बट साइकिल जब रिपीट हो रहा है तब हम वापस oxaloacetate के पास आ रहे हैं, which is 4 कार्बन. यानी कि during the cycle which is taking place, 2 molecules of carbon need to be released, and that is happening by 2 successive oxidative decarboxylation. यानी कि 2 बार आपका CO2 molecule जो है रिलीज होगा. ठीक है, क्लियर है? हाँ, this step.
Yes, ma'am.
So this is important like, to understand कि क्यों हमने 2 successive oxidative decarboxylation बोल रहे हैं. So the cycle needs to be continued. तो इसलिए जब आपका ये 6 कार्बन बन जाता है सिट्रिक एसिड, इसको दोबारा यूज करके oxaloacetic बन गई 4 कार्बन, तो आपको -2 कार्बन करना है, और वो कैसे होगा आपका oxidative decarboxylation step से. Okay. Now, in this reaction, alpha ketoglutarate is oxidatively decarboxylated into C4-thioester called succinyl CoA, and U2 is released. Okay, again delta G is negative. So this reaction is virtually identical to the pyruvate dehydrogenase complex reaction. In both, it promotes oxidation of a keto acid with the loss of carboxyl group at CO2. Okay. यहाँ पे जब हम बोलते हैं डेल्टा जी नेगेटिव है, इट इज स्पॉन्टेनियस. जब आपको डेल्टा जी पॉजिटिव होता है तब आपको एनर्जी लगती है. एंड हमेशा याद रखो हम पीछे साइकिल पढ़ रहे हैं, सो वी आर गोइंग टू जनरेट एनर्जी, वी आर नॉट गोइंग टू यूज एनर्जी. So this step is irreversible. It also produces NADH, and NADH is equivalent to 2.5 ATP, right? Okay. So the alpha ketoglutarate dehydrogenase complex shares similar properties with PDC. PDC क्या है आपका pyruvate dehydrogenase complex. Again, it contains similar to PDC. यहाँ पे भी आपके क्या थे 3 subunits है, यानी 3 enzyme components है. Same way, आपका alpha ketoglutarate is also having 3 enzyme components. And what are those components? Alpha ketoglutarate dehydrogenase or decarboxylase, transsuccinylase, and dihydrolipoyl dehydrogenase. Okay. So if you have understood how PDC works, then it is easy to understand how alpha ketoglutarate dehydrogenase complex is going to work. Okay. So I'll be sharing the PPT with you guys so that you can go through and understand how many पहले ले चुका है about the pyruvate dehydrogenase complex. Again, similar to PDC, it also requires 5 cofactors as which is required by the pyruvate dehydrogenase complex. क्या क्या हो गया आपके thymine pyrophosphate TPP mention होगा, lipoic acid, flavin, adenine dinucleotide, normal जिसको हम FAD बोलते हैं, coenzyme A and nicotine amide adenine dinucleotide, ये हो गया आपका NAD. ठीक है, इतना क्लियर है स्मृति? ये स्लाइड आपको क्लियर है?
Yes, ma'am.
Okay. बहुत कुछ है इसमें याद रखने को. That's why I'm going very slowly, and it is really important क्योंकि हर एक sentence is equivalent to 2 mark question in the exam.
Yes, ma'am.
Alpha ketoglutarate is becoming succinyl CoA. Okay, structure भी आप देख सकते हैं इसमें कहाँ पे changes आ रहे हैं. So you can see that over here you are just adding the S-CoA enzyme. तो ये आपका succinyl CoA बन रहा है. Now, conversion of succinyl CoA into succinate. Succinyl-CoA is a high energy compound like acetyl-CoA. Now क्यों हम इसको बोलते हैं high energy compound? Because on the hydrolysis, okay, जैसा G जो है आपका highly negative है, यानी कि जब हम यहाँ का bond तोड़ते हैं, succinyl-CoA का जो bond तोड़ते हैं हम लोग यहाँ पे, it releases tremendous amount of energy, and that's why it has got high negative delta G upon the hydrolysis. So this can be coupled to directly phosphorylation of GDP to GTP. यानी कि क्या इतनी एनर्जी हमें मिल रही है कि मैं GDP को GTP में कन्वर्ट कर रही हूँ, and GTP is equivalent to one molecule of your ATP. यानी कि क्या at the end of this I have generated another energy molecule. ठीक है? Now this is an example of substrate level phosphorylation. The energy released during the hydrolysis of the hyoester bond of succinyl-CoA, is accompanied by phosphorylation of GDP to become GP. Okay, and of course this enzyme is catalyzed by the succinyl-CoA synthase. जब भी synthase हो या synthetase हो, always remember यहाँ पे GTP या फिर ATP आपका use या involve होगा. Clear? हमेशा याद रखो synthase, synthetase है तो यहाँ पे GTP या ATP आपका इन्वॉल्व जरूर होगा. डिहाइड्रोजनेज है तो आपका NAD इन्वॉल्व होगा. Now the GTP readily donates its terminal phosphate group to ADP to form ATP by the action of NT2 plus dependent enzyme. अभी हम even हम बोलते हैं कि GTP बना है, it is equivalent to ATP, but it is not direct. Okay, this is how it is forming. कैसे? The phosphate group from the GTP is transferred to the ADP to become ATP, and this is done by the enzyme called nucleoside diphosphokinase, which is present in the interspace of the mitochondria. Now let's see the reaction over here. This is the reaction. So GTP plus ADP is going to give you GDP plus ATP. इसलिए हम बोलते हैं कि TC cycle में जो एक GTP मिलता है, it is equivalent to one ATP. Now it is clear why it is equivalent to ATP, because immediately once the GTP is formed, it donates its phosphate group to the ADP with the help of the enzyme called nucleoside diphosphokinase with the cofactor ND+. Clear?
Yes, ma'am.
Now let's talk about the similarly over here. ITP यहाँ पे क्या? Inosine triphosphate. Inosine. जब हम लोग न्यूक्लियोटाइड सिंथेसिस पढ़ेंगे तब उसमें ये आइनोसिन, हाइपोक्सांथीन, xanthine ये सब आएगा and we'll study over there in detail. Now let's talk about the dehydrogenation of succinate to fumarate. So the oxidation of succinate to fumarate is the only dehydrogenation reaction in the PCA cycle in which NAD+ does not participate. Okay, in this reaction, hydrogen is directly transferred from succinate to flavoprotein enzyme and then the fumarate is formed. Usually क्या होता है आपका succinate से directly fumarate बनना चाहिए. यहाँ पे क्या हो रहा है? Succinate dehydrogenase करेगा तो यहाँ पे आपका NAD+ इन्वॉल्व होना चाहिए. अब यहाँ पे क्या है? इसके थ्रू ना मतलब डायरेक्ट substrate to product ना जाके they are making use of the direct transfer of the hydrogen from succinate to the FAD. Okay. If you remember, from succinate to fumarate, we have got FADH2 release. ठीक है? अब यहाँ पे क्या हो रहा है? Instead of substrate to product बनने में, substrate से जो आपका H आएगा वो डायरेक्टली हम यहाँ पे फ्लैगेंट प्रोटीन में देंगे and आपका fumarate देंगे. So succinate dehydrogenase is a flavoprotein which is located on the inner mitochondria. Okay, and it has got a prosthetic group of FAD as the coenzyme. So it acts as the hydrogen accepting the reaction rather than NAD+. इसलिए TC cycle में अगर आप देखोगे, so we have got 3 NADH, 1 FADH2 and 1 GTP. अब FADH2 आपको कहाँ बन रहा था? इस स्टेप में जहाँ पे सक्सिनेट टू फ्यूमरेट होता है with the help of the सक्सिनेट डीहाइड्रोजनेज. तो यूजुअली मैंने क्या बोला हमेशा, जब भी आपका डीहाइड्रोजनेज होता है आपका NAD+ इन्वॉल्व होता है, राइट? दिस इज द ओनली केस जहाँ पे आपका इंस्टेड ऑफ NAD+ इट इज FAD. ओके, याद रहेगा स्मृति?
Yes, ma'am.
Okay. So in succinate dehydrogenase, isoalloxanide ring of FAD is covalent linked to the histidine side chain of the enzyme, and then the reaction is reversible reaction. Finally, what is happening? Fumarate आपका बन गया है. A fumarate जो है, it is hydrated to form malate in the presence of the fumarate dehydratase or fumarase. Simple, fumarase. This is a reversible reaction and involves hydration in malate formation and dehydration in fumarate formation. अब एक बार आपका malate बन गया है, तो malate dehydrogenase क्या करेगा? It is going to convert into oxaloacetate. Simple. Okay, so fumarate hydratase is highly specific and it catalyzes trans addition and removal of HOH and does not act on the malate. This is isomer of the fumarate. Now, malate dehydrogenase is a good example of a reaction which has got net flow opposite to an unparalleled equilibrium. यानी कि क्या? It is delta G आपको यहाँ पे positive है, okay, and that's why यहाँ पे आपका oxaloacetate जो बन रहा है, फिर उसमें आपका NADH आपका use हो जा रहा है in order to make it favorable. Okay. Okay, स्मृति, हमने भी सारे स्टेप्स यहाँ पे कवर कर लिए हैं. आपको लगता है कोई स्टेप ऐसा है जो डाउट में है या कोई स्टेप समझ में नहीं आया है?
Ma'am, I need to go through the literature once. Then if I have any doubts, definitely
Yes.
मैं approach करूँगी आपको.
Yes. So what you need to do first remember, try to remember the entire cycle and then read all the steps. कि कब कौन सा enzyme कैसे act कर रहा है. So once you remember, like citrate के बाद आपका cis-equanitate आता है, उसके बाद आपका alpha ketoglutarate आता है. अगर एक बार ये याद हो जाएगा, then it becomes easy to understand the entire cycle. Okay. Yeah.
Yes, ma'am.
So just let me know if any of the doubts are there. Okay. So thank you for listening, and let me know if any of the doubts are there.